All right. Yum, yum, yum. Let's see. Hang on, it's back here. Big view. All right. So anyway, um, essentially what we're going to be doing today is giving you some in-depth overview of PCR, which is a very powerful technique used in molecular biology. And not super, super old. I mean, it is relatively old, I guess, probably compared to any of us. But in terms of science, it's still fairly new. Then essentially giving you a heads up as to how we're going to use PCR to amplify the section of the yellow gene that we're interested in, and then use that to see how genotype equals phenotype, essentially, right? So at the end of this, you'll have a comparison between, pardon me, as I'm at lunch, between wild type copy of the yellow gene, or at least fragment of it, and the mutant copy, which results in yellow flies. Okay, so we're going to bounce around a little bit just because it's kind of like a non-linear topic. But the general principle behind, hang on, how do I get? Yeah, sorry, I'm just trying to see how. What does that do? No, I don't want that. I really don't want that. I'm just trying to make myself bigger. Well, anyway, the idea behind PCR is that if you want to do any work with DNA, or even with RNA in a sense, right? But we're not going to really cover that today. But if you want to do any work in terms of molecular biology, and that could be a lot of different things, sequencing stuff, just see what's there, what it is. Could be cloning a gene in order to express it in a different system. Insulin actually was the first example of that before molecular biology allowed it to be cloned into bacteria, or was it yeast? I'm not sure which. Essentially, the only insulin that we had for diabetics came from pigs, which isn't good for pigs, obviously, because they aren't around anymore. Because we need to get their pancreases out. It's also super, super expensive to do it that way, and you expose yourself to kind of zoonotic diseases, things like that. So that's just one application for PCR essentially. Lots and lots of other applications. I mean, really, it just depends on what you can think of, right? And the big thing that PCR solves is quantity, right? So for every cell in your sample, whatever that may be, muscle tissue or bacteria or yeast or whatever, each cell, if it's a eukaryote, has 2 copies of that gene, right? Whatever you might be interested in. And so even if you have millions of cells and your DNA purification is perfect, neither of which may be the case, you're only going to get a few million copies of that gene, which may not be enough to do anything with. Also, another issue with any kinds of molecular biology is that you really want to work with what you want to work with, not with the entire genome of an organism. So, you don't want all that other crap. Well, it's not crap, that's kind of pejorative, all those other genes and stuff. And also you want a lot of the thing that you do want. And so PCR is really the kind of way of doing that. And so I'm going to walk you through the general principle, give you a little bit of history, which is fun, and then we'll see how that applies to the project that we're going to be working on. Okay. Get whiteboard out here. So whenever you start with any kind of PCR, you have to figure out, well, what bit of DNA do I want to amplify? And so, right, so this is a bit of DNA, as you can probably tell. and found the hole in my board. This is the gene that you're interested in, let's say, right? So this is just an example. So this is our gene of interest, or GOI. And we want to get that out of its native environment on that chromosome, and we want a whole load of it to be able to do whatever we want with cloning or sequencing or the like. So the very first step you need to do, let's tilt this a little bit further this way, is to design what are called primers, right? Primers are, if I can spell it, single-stranded DNA matching your target, right? And so typically you need to know at least something about the sequence of your target. There are other ways of doing it without knowing that. They're a lot clunkier and take longer. But typically you're going to know what you're heading after, maybe because it's you're using a related gene in another organism, maybe, or because you have the genome sequence. You know, there's a lot of different ways of doing it. But essentially you take this sequence here and you go, okay, I want to clone all of this. I don't want to clone over here. I don't want to clone over here. I want just this bit. So I'm going to design my primers, and you have the DNA sequence, so you know what it's going to look like to this way, and because you need to have the whole caboodle that way, right? So this is called your forward primer. And this is your, bloody hell, reverse primer. Can you all see me large enough? Because I is tiny on my screen, I can't fix that. Austin's huge for some reason, but hopefully it's all nice and big for you. So essentially what's good, this is the first step, right? And these are designed to base pair with the respective strands. We typically write them like this, but really they're actually matching up here, right? The opposite strand. You don't have to worry too much about that. It doesn't really matter, to be honest. And whenever you do design these primers, you always just use a piece of software that goes, I want these primers, and then it will tell you the sequence, typically. Anyway, so they match and will bind with, and they're very specific to this bit of the DNA sequence and to this bit of the DNA sequence. And so if you remember from DNA replication, right, way back in the day, you remember that this is what also happens in DNA replication, right? Primase or DNA polymerase alpha makes, in that case, an RNA primer to serve as the basis for extension of that template, right? We're not going to use RNA primers because, you know, they're not going to hang around for long enough. And yeah, we just don't want to. So we use DNA primers, but it's essentially the same principle, right? It's to provide a 3' OH, tiny, for DNA polymerase, and we'll talk a bit more about that in a second, to start extending that primer into this sequence, right? So the primers are what give us our specificity, right? Because they're usually long enough, around 20 to 25 base pairs long, that the chance of them accidentally binding elsewhere in the genome is pretty much close to zero, right? And you can design them in a way to make them more or less specific if you wish, right? So that's the first step. And then what we're going to do is we're going to mix these primers. So we're going to have primers, we're going to have DNTPs, right? Those are basically your ACG, yeah, that's right. Your ACGs and Ts, right? Because you need that to kind of make more DNA, essentially. You need what's called TAC. DNA polymerase. I'll get to why it's called TAC in a little bit. That's your, I don't know, DNA maker, basically. What else do you need? You need buffer. And that basically makes TAC, oops, happy, right? Puts it in the correct kind of chemical mixture, essentially, I guess, you know, kind of trying to replicate a little bit of what it's like inside the bacterial cell. And then you have magnesium. I think we talked about this briefly another time, where enzymes needing cofactors, right? Magnesium is the one for TAC. If you don't put it in, it doesn't work. It's as simple as that. And you can actually use the concentration of magnesium to make TAC more or less specific, which is kind of funky. I don't really understand how that works, but I know that it does. And so you can, if you're troubleshooting a PCR, one of the first things you troubleshoot is magnesium concentration. And that's very much specific on your gene of interest and your primers as well. This PCR, I lay you on a secret, it's black magic, right? We pretend to know how it works and we act all knowledgeable and everything, but really it's, yeah, it's magic, essentially. Because you put all this stuff in a tube, mix it together, and you put it through the appropriate temperature cycles, and then maybe, if you're lucky, it works and you have a nice band. If it doesn't, you're like, oh shit, why didn't that work? And then you have to go through a whole bunch of troubleshooting. And actually, yeah, I think it was Penn State, there was a rubber chicken which was hanging above the PCR machines, so that every time you set up a PCR, you'd shake the rubber chicken and that would help it work. I'm not joking. I don't know if it, I mean, we didn't do like a double-blind trial to see if it really did help, but just kind of give you an idea of how mysterious this process can be. And honestly, sometimes it works great. In fact, yeah, totally superstitious. I'm usually very sad when it works perfectly the first time for a student, because then they just think it's going to work like that every time. It's like, no. Sometimes it just doesn't work for no apparent reason. Don't know why, right? So if it works first time, they get all their hopes up and they think it's going to be like that forever. And it's like, don't want to give you like a misleading sense of optimism or anything. I mean, I've actually spent, months in one case trying to get a PCR to work. Other times it works first time. So anyway, you mix all that stuff together. Typically we make what's called a master mix. This is all in the handout as well. Simply because some of these things are very hard to measure accurately. I was going to tell you why TAC is called TAC. So this is kind of fun. I always like stories about how scientists kind of go against the grain and break dogmas, right? Because in my mind, dogmas are kind of a stifling concept, essentially, right? It's always got to be this way. And it's like, well, no, it doesn't actually have to be. And so back in up to the 60s, give or take, yeah, maybe early 70s, one of the dogmas of biology as a whole was that life could not live above 60 degrees Celsius, whatever that is in Fahrenheit. Big number, I guess. Because proteins fell to pieces, right? They denatured, right? Which is a fancy way of saying falls to pieces. So what happens when you fry an egg? right, that translucent egg white actually turns solid white and hard, right? That's because of the albumin protein denaturing with heat and then basically reforming into the incorrect form of the protein, right? So that's why when you fry an egg, that's why your egg white is nice and hard and white, and it's not goopy and clear. Anyway, so up until that point, PCR had actually been invented in the 60s-ish, but we had to use like normal, what we call mesophilic bacteria, like E. coli. It grows at 37 degrees Celsius, like our cells do. And that causes all kind of problems for accuracy, which we'll get to maybe in a little bit. And also, it's really tedious as well. They actually had like individual temperature baths, and the scientists doing the PCR had to kind of take it from one bath to the other like 35 times. I'm really, really glad that I started my career after that process. Where we go. So, get out of there. One cool invention was something that's called a thermocycler or thermal cycler. I call them thermocyclers. And essentially what these are a very fancy kind of like mobile fridge like you have in your car. I got one in the back of mine for storing water and stuff. So you know those little portable 12-volt fridge thingy, cooler things? Well, they've got something in them called a Peltier, which is a way of controlling temperature. In that case, it's to keep things cool. And so thermocyclers have one, but it can vary in between different temperatures very quickly and reproducibly. So that made life a lot easier. And we'll talk about what temperatures those are in a little bit. However, we still had the problem of having to use a DNA polymerase with a very low extension temperature, like the temperature at which it works. And that all changed in the early 70s by, I think it was people from the University of Idaho, maybe, not 100% sure on that. And essentially, they did a whole bunch of work here. Anybody know where this is, just out of curiosity? Some of you may have even been there. This is a place called Grand Prismatic Spring. Actually, that's my youngest daughter. She's probably going, Daddy, I don't want to fall in and boil alive or something like that. I don't know. Yellowstone, yeah. So this was when my family and I moved from Pennsylvania to Washington State. And so on the way over there, we didn't have cats at the time. We actually stopped at Yellowstone just for a day, unfortunately, as part of like a pilgrimage, right? Because, I've been a molecular biologist for decades, basically. And all of these different colors that you can see here. So this is steam because it's hot. I don't think this one is crazy balls hot. I think this is only, I don't know, like 80 degrees Celsius, give or take. But all of these different colors that make Grand Prismatic Spring so pretty, and a lot of the springs in Yellowstone, are pigmented bacteria or archaea. It kind of varies. And so there are microorganisms living in these springs. Hikers don't tend to do very well. They tend to die. But bacteria thrive pretty well. And what these researchers found was that there was actually life in these springs, and it lived, I think, first one was around 70 degrees Celsius, and the limit has just gone higher since then. I think the current high temperature limit is 115 degrees Celsius. And those are archaea that are found in black smokers, basically high temperature seeps in the Earth's crust underwater. Anyway, so they they isolated bacteria, and a bunch of people said, no, that can't be true. Yeah, hypothermophile, that's the one. Actually, this is one of my favorite species names. Pyrococcus, oops, if I can spell it right. Furiosus. Furio. There you go, something like that. And that's the current champ at, I think it's around 115 Celsius, give or take. And so the reason why it can be 115 Celsius, which is technically above boiling point of water, is that they're very deep beneath the ocean. So pressure is up, and so temperature can go up without the water boiling, essentially. Anyway, so that's the current. Oh, and Pyrococcus furiosus means angry, flaming balls. I think microbiologists like get all the best species names, in my opinion. Coccus is a ball or sphere. Pyro, fire. Furiosus is angry, essentially. But anyway, the ones that they isolated were a little bit more. Those are actually typically just actually thermophiles rather than hypothermophiles. And they named it Thermus aquaticus. Sounds kind of like something out of a ancient history, like Roman story or something. I introduced Thermus aquaticus from Rome or something like that. I don't know. Anyway, that's basically hot water loving bacteria. And that is shortened because whenever you get enzymes from bacteria, you take the first letter of their genus and then the second and third letter of their species, or first and second letter of their species name. And so that is shortened to TAC. And that is the enzyme that is used in PCR. And so the reason for using TAC is that it works at 72 degrees. And by doing so, it allows the PCR to work at basically temperatures which ensure the specificity of the primers. And so primers are based on complementarity, and they have what's called a annealing temperature, or which is actually a melting temperature. So above that temperature, they kind of fall off unless they're really, really well stuck. And so if you use a low temperature enzyme, then it's a very permissive temperature, right? So it means that those primers can stick elsewhere where they're not supposed to. Whereas if you use TAC, then they have to be perfectly matched. Otherwise, the bonds holding the primer to the DNA are not strong enough, and the primer falls off. And so that was actually one of the biggest discoveries probably in the history of molecular biology. I think TAC is, or Thermus aquaticus, without it, we wouldn't have the human genome sequence. We wouldn't be able to make transgenic plants or animals. We wouldn't be able to clone insulin, right? Pretty much all of molecular biology would not be as it exists, which is essentially an awful lot of medicine as well. A huge amount of medicine, in fact, ever more so, right? So it's a little hard to overstate how important this discovery was in terms of opening up molecular biology. And really it took off like crazy after that, right? And the invention of the thermocycler as well, which I think was by Roche, French company. They minted a lot of money from that. Anyway, so this is Grand Prismatic Springs, super pretty. If you ever get a chance to go to Yellowstone, and it's a fair way from here, no, but if you ever get a chance to go there, you should grab it with both hands because it is a stunningly beautiful place, right? I mean, it's a beautiful part of the world anyway, but it's like being in an alien landscape. It really is. So if you ever get the chance, give it a shot. And if you do, try and spend more than a day there like we did, because you could easily spend a week there and not see half of it. It's an enormous place. Lots of bison as well, which is really cool. Anyway, so I need to see that no more. So basically what we're going to be doing in PCR is we need to get those primers to stick. Well, we need to open up our DNA first. We need to get our primers to stick. We need to extend our new sequence using TAC. And then we just repeat the same thing over and over and over again. And so really it works like so. So if we go back to our gene of interest, like so, right? This is double-stranded DNA. So our primers can't bind here, right? They need single-stranded DNA to bind to them, right? So the first step, the very first step of all of any PCR is called denaturation, right? And so I'm going to do that in shorthand. I'll probably better not. All right, I'm full. And that's usually around 94 degrees Celsius, give or take, right? It doesn't really matter. Basically, you're putting enough thermal energy in here that all of the hydrogen bonds between those two strands break. And so what that gives you is that. Maybe not for all of it, but for enough of it. Single-stranded DNA. And so now that you have single-stranded DNA, now your primers are able to bind. This is called the annealing step, which is weird because it has a very different meaning in metallurgy. But in PCR, at least, it's the temperature at which these primers will stick here and nowhere else, right? And so this is a variable, right? Depends on the primer length and magic, basically. And so this is typically 60 to 65, or maybe 55 actually. 55 to 65 degrees Celsius. Yeah. Again, this is a value which is both theoretical, because it's based on the sequence of the primers, and empirical as well, because that temperature might not actually work. So you do sometimes have to do a lot of guessing and experimentation, I guess, to find the right temperature. And then what we're going to do is we're going to add our, well, we don't add it, it's already there, but we're going to raise the temperature to the level which our DNA polymerase starts working. Right, and so let's draw this out again. I gotta do some different colors. Right, so there's our TAC, and what TAC's going to do is it's going to run for as long as it can, basically. So it's going to go chink. The sounds are really important. Actually, if you listen closely, it's just like that. And so our TAC is just going to go, and you notice here, right, it's gone past the end of the bit of DNA that we want. That's not an issue. And so this is this is the extension at 72 degrees Celsius, right? So those are the basic, trying to make sure it's all kind of lined up right. Basic phases of PCR. Denaturation, to melt the DNA so it forms single strands. Primer binding, which is one hopes at a temperature which is specific to this gene, so those primers don't bind anywhere else, giving you like off-target products, which is a real pain because then you have to figure out which is the right one. And then once that's happened, extension, which is where our TAC comes in and basically uses this template strand to make another copy, right? And so at the end of this is one cycle, right? Out of usually about 30 to 35, depending on application. And so what happens now is that we have I should have left more space. We have two copies, right? So we started one copy, go through a full cycle. Now we have doubled our copies. That doubling is important, right? And I'll get to why that is in a second. Let me just take a picture of this. Anybody got any questions so far? If you do, pipe up, write them down. All right. So I'm going to take this and I'm going to just stick it back up here. So now we start essentially, damn it, I should have written on the other one. Cycle 2. We have our, hang on, how did it go? It would have been, we have that, right? And that's because this is where the reverse primer was, and this is where the forward primer was, right? Again, any questions, just really please do stop me. Otherwise it feels like I'm just talking to myself sometimes. I like questions, right? We go through the same process of denaturation and annealing, right? Oops. Hang on, I've got to make these a little bit apart to show that they are actually annealed. And this new strand could go quite a long distance past where your gene of interest ends, right? So now again, we're going to get our reverse primer. We're going to get our forward primer, FR. I actually did that the wrong way around. Never mind. It doesn't matter. It does. Got to do it right. Sorry. Pedant in me was having a little fit just then. Forward primer is going to go to this one, and then reverse primer to that one. All right. Yeah, I know. I'm sorry. Just can't help myself. Right. So this is, we've already melted these strands apart. We have loads of primers, right? We supply them in huge excess. And those primers have now found their, their spots, so to speak. Let's use a different color now just to Oh no, maybe I'll use red again. And then we add our tack and we get the same thing all over again. That goes actually past that. So our tack comes in and it just does the same thing again. It just extends the primer and creates a new strand, right? Now we have 1, 2, 3, 4 copies. I'm not going to keep doing this because my arms will just get tired holding this thing up. But every time you run a cycle, at least until things start getting short supply or getting tired out, you double the amount of copies of that piece of DNA. And as you can see, even though on early stages, some of those copies are longer than they need to be, very quickly, you're going to get the actual length. There's one just here, and there's one just here, which are the correct length. And those again serve as templates for the next round of replication, and over and over and over again. And so essentially what happens is you're going to do this typically between 30 and 35 times, right? So at the very beginning, if we can plot this as a graph, right? If you have what's the cycle number and actually let's put number of copies, right, on the y-axis. You know, start off here and end up, let's say 30 will be enough, right? It takes a while to notice an increase, right? Because the first cycle is from 1 to 2, then it's 2 to 4, and then it's 4 to 8, 8 to 16, 16 to 1632, 64, 128, 256, right? It takes a while, but after a bit, it goes exponential, right? And that's exactly how we end up with basically a shit ton of DNA, right? After a while, however, though, the polymerase kind of gets a little bit run ragged by being heated up to 94 degrees Celsius a whole bunch of times. And also DNTPs may start running out, or maybe the primers, even though we provide quite an excess. And so after a while, that curve, sorry, just trying to struggle with getting the lights right. That curve kind of levels off. It's actually quite a lot similar to a bacterial growth phase, to be honest. You know, nothing very much happening down here. This is our exponential increase. And really, once you get, for example, if you get up into 1 million, then the next cycle you're going to have 2 million, and then 4 million, and then 8 million, right? So it's increases very, very quickly. And then essentially you reach saturation, and the increase in number of copies kind of levels off. But by the time you get up here into that part, you're in the millions to billions of copies of your target sequence. And so now you have a whole load to do whatever you want with, right? Whatever that might be. Send it off for sequencing, could be, if you want to clone it, maybe you just want to produce it as straight up DNA for injection as part of like a DNA vaccine, right? That's, I think, not 100% sure because I haven't read all the details about it, but the COVID-19 vaccine that's being produced by Oxford University and its partners is an RNA vaccine. So it's actually kind of like the same principle, but using RNA polymerase to generate huge numbers of RNA copies, which then get injected into a patient and produce proteins, which raises the elicits an immune response. Right. So the process is, well, the principles are the same. Process is obviously a little bit different because it's different type of nucleic acid. Right. So that's the general principle behind PCR, right? And that's what makes it so powerful, is that you can start with one copy, right? So technically, not quite practical like this, but technically you could start with one cell, right? And you can end up with enough DNA to clone something, to sequence it, to tell whether or not that person committed that particular crime. And so actually, there you go, I forgot another application. PCR is one of the breakthroughs in forensic science, which allowed prosecutors to actually, well, both exonerate and also incriminate. Not sure that's the right word. Should be, but it doesn't sound right. Basically, put the finger on someone and figure out that they did incriminate. There you go. There's an extra I and an N there. Incriminate someone, because before, you know, you'd need huge number of cells, and you'd do like kind of antibody stuff and things. But being able to use PCR means that, you know, just the like scrapings of some skin cells underneath the victim's fingernails, or some hair that was left on a victim, all kinds of things like that, really tiny amounts of genetic material are now sufficient to be able to expand that to the point where you can tell whether or not a particular individual committed that crime, or at least left that biological material on the victim. Right? Obviously, the one does not necessarily equal the other, but in many cases, obviously, it does. And so that's what makes this technique so utterly awesome, basically. Actually doing it is, like I said, is somewhat more mundane, at least after the first few times. But hopefully you'll all each get a chance to do this at a later point, you know, because it's just kind of neat, particularly when it works. Okay, so that's the basic overview of PCR. Oh, and actually this was such a breakthrough that the person who invented it, Kary Mullis, invented PCR, equals Nobel Prize. Nobel Prize in Chemistry, just to show you how important this was to humanity, right? If we want to be like kind of grandiose about it. Certainly individuals who control their diabetes, you know, whether type 1 or type 2 or whatever type they have, they would not be able to do so anywhere near as easily as they do currently, and at relatively low cost without PCR, right? Because we wouldn't be able to clone insulin and produce it in large quantities. Same for an awful lot of different things, right? So it's really hard to overstate how important this discovery was. Okay, so what are we going to be doing? Well, right? So, instead of just random, beep, all right, and I'm going to draw this out semi-accurately. Right, I think there are more exons than that, but this is yellow. All right, so this produces a protein which is, as far as we know, we don't fully understand what yellow does, even though it's been studied for not hundreds, but at least 100 years, over 100 years actually. It's involved in mating and pigmentation. right? The pigmentation bit is obviously the basis of the yellow phenotype. And what we're going to be doing is we're going to amplify a part of this. We can't amplify all of it because it's too big. One of the limits of PCR, depending on which type of tack you use, actually there is a tack from Pyrococcus furiosus. It's called PFU, just as a little bit of random information you don't need to know. Which type of tack you use determines basically how big of a piece of DNA you can amplify, because they have, I don't know, there's trade-offs essentially, right? The longer and more accurate the piece of DNA, the slower it happens, right? So the harder it is to get enough DNA to do stuff. And so we can't amplify all of the yellow gene, right? Typically, if you wanted to clone it as is, you would clone different chunks of it, right? You wouldn't try and do it all in one go. And so what we're going to do, probably should have made this a bit bigger. What we've done is we've designed our primers to just the start codon. Oops, that's a T. It's not RNA yet. To the just before the start codon, because that's pretty sure where the mutation is that we're interested in. To, I think it's the junction between exon 1 and intron 2. Not 100% sure. I'd have to look at an alignment to jog my memory. So we're going to only amplify basically this little bit. That's AUG, not AUF. Oof. There you go. So we're just going to amplify this little part. Part of the reason is that the type of sequencing that we do won't sequence more than like 600 to 700 base pairs accurately. So you're limited anyway. There's no point doing all of this because you're not going to be able to sequence what's in the middle. And that is based on our primers design, at least 401 base pairs, right? I don't know how big the whole yellow gene is, but it's most likely in the thousands of base pairs going on just normal eukaryotic genes, right? So we're only going to be sequencing, that's a T. Keep thinking about RNA. We're only going to be amplifying and then sequencing a small chunk of that, right? Because we don't need to do anymore because it's essentially that spans our region of interest. Now there are alleles, right, or mutations all through the yellow gene. There's at least hundreds of them. And we're not going to be able to detect those. But we're pretty sure, based on the flyers that we ordered from Carolina Biosciences, that the mutation that we're interested in is in the start codon. I think it's a G to C transition. I'd have to double check from memory. So when we sequence our flies, when we sequence the wild type flies, this is a sequence we should see. When we sequence the yellow DNA from the yellow flies, we should see a difference in the start codon. Essentially, it's no longer going to be a start codon. And that's pretty much it, I think. So typically, once we do this, we take some of that PCR, right? Because it should have, a huge amount of DNA in it. We don't need to run all of it. And we're going to run a very small amount of it on a agarose gel. So again, like we did with the DNA QC, right? We need to visualize the DNA. And so we're going to run it on a gel, which is basically what the next lab will be about, and make sure that it is the right, A, it's there, right, which is important, and B, that it's the right size as well. And then once we've done that, then we can clean it up, we can figure out how much we have, how pure it is, essentially a repeat of the DNA QC process, right? Because we don't want to ship this off to a sequencing facility if it has leftover primers and Taq polymerase and things like that. We want to make sure that it's just DNA and nothing else for them to add to whatever they need to add it to get it to work. And I think that's pretty much the process. Let me just double check, make sure I've covered everything. Again, check out the lab handout. So the assignment, which should be available already in Blackboard in the folder for this lab. The assignment will be, again, asking you kind of basic questions about the process and various little details and things like that, what we talked about, and also about some of what we talked about with the yellow gene too. And that's pretty much it. Whew, I'm all tired out. I get very enthusiastic when I talk about PCR. So I cloned my first gene. When was that? That was a summer project between summer of 1998. Yeah, summer of 1998 and 1999. Yeah, no, summer of 98. There you go. Summer can't be between years, that's winter. So summer of 98 was the first time I cloned a gene, which was from, oh man, I can't remember what the species is anymore. Type of bacteria that causes food poisoning. Can't remember what it's called. Not salmonella, wasn't anything that dangerous. But yeah, so that was 22 years ago. Kind of crazy to think about it. And done an awful lot of other different funky things with it in the process from there until now. I don't tend to do much PCR or cloning anymore because most of my my research tends to be using existing materials and tools, right, which I don't necessarily need to make myself. But to finish off a current project, I'll probably need to make some cell-specific expression vectors for a gene in C. elegans, so I can turn on or turn off, depending on whether it's by RNA or by expression, a particular gene in a certain group of cells and not in another group of cells. So I can look at cell-specific expression and function. So that'll be on the list at some point, I presume. And that's pretty much it. So if you got any questions, give me a shout. Hopefully it's all fairly self-explanatory. Pictures are going to go up to Blackboard as soon as I have finished with you lot today. Yeah, and it will be due by midnight next Tuesday. So we have time to talk about any questions you may have next Tuesday during lab as well. That's it. Anybody got any questions about this lab or last week's? Awesome sauce. Well, thank you, everybody. Have a lovely weekend and see you next week. And good luck in your exam.